It's really important if you're trying to understand and generate a complete spatial biology solution. There's actually two distance scales that are really important. We have two platforms that cover the two distance scales. The GeoMx is a platform that really monitors what I call molecular machines in tissue that do work. These are aggregates of cells that range from about 50 microns in size to about 2 millimeters in size. And that's the specialty of the GeoMx, the functional in situ behavior of these molecular machines that accomplish a task in a tissue. Now, when you want to take that task and you want to go and do what I call understand the private lives of the individual cells and how they talk to each other at the single cell and subcellular manner, that's why we invented and developed the CosMx Spatial Molecular Imager. So the CosMx Spatial Molecular Imager would allow you to tell what all the little private conversations between the individual cells that will be organizing to make a function, how they talk to each other, how they communicate. But then when you want to understand how they function, then the GeoMx whole transcriptional profile tells you how the whole thing works. So that's how these two platforms work together. I sort of view it like it's an analogous. It's like if you're interested in like Formula One racing, say, the race car on the track is GeoMx. We measure that. Whereas if you want to take the car into the garage and understand how that little Formula One car does, then you get to use the CosMx to take it apart bit by bit. But you can't understand how a race car functions without taking it on the track. So that's sort of the CosMx is how it works, and the GeoMx is how it functions. Those are the two things. It's absolutely true. The spatial biology revolution exists because all life on this planet, as far as we can tell, lives in three spatial dimensions, X, Y, and Z. And the way the cells are organized, the ways the tissues work together is all a spatial organization game. So the fact is we've been ignoring that for literally centuries. And now finally we're at the place where we have the tools to understand how nature really designed things to work in space. And that's really what I call rewriting the textbooks of life sciences. We're now at the place where we can take every field that used to not be a molecular field and turn it into a spatially resolved molecular field. Developmental biology, plant sciences, cancer immunology, developmental biology, all these things are ripe for the spatial biology revolution. And that's why people call it the spatial biology revolution, because it will allow us to rewrite the textbooks.